Hey guys, in this video we are going to be going through secondary hemostasis physio. And if you recall from the video on primary hemostasis, we talked about how primary hemostasis was development of a platelet plug. Secondary hemostasis is the clotting cascade or the coagulation cascade. Now, secondary hemostasis again is the clotting cascade, and remember, all clotting factors are negatively charged. This is important whenever you consider how primary and secondary hemostasis work together, because again, recall how in primary hemostasis, calcium, which was positively charged, vasoconstricted, but it also attracted negatively charged clotting factors. And this is what we're talking about here in secondary hemostasis. Now, secondary hemostasis consists of both the intrinsic and the extrinsic pathways. And keep in mind, I've seen a lot of photos of the clotting cascade or the coagulation cascade, and it will have these Roman numerals of factor 12 goes to factor 12A, which goes to factor 11, which goes to factor 11A. I'm not going to overcomplicate this, okay? Whenever I first started learning the coagulation cascade, it seemed very intimidating. However, by simplifying the image in and of itself, it can really help us understand it more and just conceptualize more easily. So just keep in mind, I'm writing the actual numbers and I'm writing an A beside it to remind you that this is the clotting factor in its active form. So the intrinsic pathway starts with factor 12A, which will go to factor 11A, which will go to factor 9A, then to factor 8A. This is the intrinsic pathway. The intrinsic pathway will meet at the common pathway with factor 10A. Okay? Factor 8A has cofactor of VWF, as well as protein C and protein S. Now, the extrinsic pathway will start with tissue factor and will go to factor 7A, which will meet in the common pathway with factor 10A. Now, I like to draw factor 5A here with factor 10A. Again, it's only for simplicity's sake. Factor 5 also has VWF as a cofactor, as well as protein C and protein S. Keep in mind that protein C and protein S work with factor 5 more. However, they still work with factor 8. So again, 5 more so than 8, but both of them. Now, factor 10A, again, this is the common pathway, 10A and 5A. 10A will go to factor 2A, which will go to factor 1A. And with the help of factor 13A, we will form a stable fibrin clot. Now, although I, again, did not want to overcomplicate things by drawing, the inactive form and the active form, the only two places where I think that this is really important to know the inactive form versus the active form of the clotting factor is here with factor 2A, because factor 2 is called prothrombin, and factor 2A is called thrombin. So prothrombin is the inactive form of thrombin, and factor 1 is called fibrinogen, and factor 1A is called fibrin. Now, these terms are used interchangeably. Factor 1A can be referred to as fibrin. Just keep in mind that fibrin is factor 1A from your clotting cascade, okay? Now, there are a couple of things that I want to add as far as physio goes. Keep in mind, what's the goal of the clotting cascade? Well, it's to create a stable fibrin clot, right? However, this clotting cascade must have a means of checks and balances, right? We don't want blood to be too viscous. We don't want blood to be too thin. So this clotting cascade has to have a way of self-regulation. So keep in mind that protein C and S will bind to factor V and inhibit factor V from stimulating the common pathway and creating that stable fibrin clot. As well as over here, protein C will bind to factor VIII and inhibit factor VIII from stimulating the common pathway via the intrinsic pathway. Now, there is only one reaction that can actually bust up clots, okay? Other inhibitory mechanisms can prevent further clots from forming. However, plasminogen to plasmin, with the help of TPA as the catalyzing enzyme, it can actually bust up a stable fibrin clot. And this is going to be very relevant whenever we talk about pharm pertaining to the coagulation cascade. Okay. Now, the other checks and balances would be antithrombin 3, which inhibits thrombin directly. Go figure, right? Antithrombin. So it inhibits thrombin, but it also inhibits 10A as well. Okay. In the next video, we are going to talk about the pathophys pertaining to the clotting cascade, and it's going to be very important for you to remember the lab values pertaining to each pathway whenever we get there. So keep in mind that the intrinsic pathway, and a way that I remember this is, do you play table tennis inside or outside? Well, we play table tennis inside. So PTT is the lab value that will be used to determine if the intrinsic pathway is functioning optimally or if it's not, right? And then the extrinsic pathway, you play tennis outside. So PT is used to determine if the extrinsic pathway is functioning optimally or not. Now, again, when we get to pharm, we'll talk about these lab values and how they can be increased or decreased dependent upon medications. But from a physio perspective, if PTT is elevated, that means that we have an issue in the intrinsic pathway. And if PT is elevated, that means that we have an issue in the extrinsic pathway. So if PTT is elevated, we need to keep in mind that there's something wrong with 12A, 11A, 9A, 8A clotting factors. And if PT is elevated, there's something wrong with tissue factor or 7A. Okay, this is a way to kind of widen the concept before we get into the pathophys. So if you found this video helpful, please like and subscribe. Thanks.